Hello, everyone. Welcome and thank you for joining our webinar. We'll be exploring biological validation results for AI-based in silico techniques in AAV development. My name is Angela Anderson. I'm the Senior Director of Marketing at FORMbio, a life sciences-focused company with software and AI-based solutions dedicated to accelerating gene therapy development. I'm thrilled to introduce our FORMbio presenter, Dr. Anusha Siriraman, Life Sciences Insights Lead at FORMbio. You can expect a 20-minute presentation on the latest biological study results from FORMsightAI models conducted in collaboration with independent lab partners. We'll host a short Q&A at the end of the session. So if you have any questions during the webinar, please type them into the chat window and we'll address them during the Q&A. So take it away, Anusha.
Hi, everyone. Thank you for the introduction, Angela, and thank you all so much for tuning in to the webinar. Today, we're going to walk you through our latest lab results that show how FORMsightAI, our proprietary AI/ML model, accurately predicts truncation outcomes and generates vector designs that improve manufacturability and safety of gene therapy products. Firstly, I'd like to provide you with a brief introduction to FORMbio and who we are. FORMbio provides computational life sciences solutions to accelerate cell and gene therapy development from discovery to the clinic. AI models and in silico techniques are already being very heavily utilized in the early drug discovery, target identification, clinical and commercial stages. But there remains a critical and lengthy development stage between discovery and clinical phases that is yet to be fully addressed by AI models. FORMbio is exclusively focused on this development stage with the aim of helping your team advance therapeutic products and as your full service in silico partner. Just like how you'd engage with a CRO or a CDMO, we think of ourselves as a contract computational organization. We're focused on saving your company and individual therapeutic programs months and millions on the way to an IND. We're engaged with many leading companies in the CGT and AI space, including academic and commercial partners. We have a variety of strategic alliances, including Google as their exclusive partner for HCLS bioinformatics platform, as well as PacBio as their hi-fi sequencing partner, and Ginkgo Bioworks for AAV gene therapy design and development. Comprised of distinguished experts from various life sciences domains, our scientific advisory board is poised to provide invaluable strategic guidance, leveraging FORMbio's advanced AI models and in silico techniques. Coming to FORMsightAI, it is a validated AI/ML technology that's developed by FORMbio that can perform in silico assessments of immunogenicity and manufacturability, enabling you and your team to optimize and de-risk your vector sequences. FORMsightAI addresses common issues faced by cell and gene therapy developers of construct truncations and manufacturing contaminations, which are major sources of unacceptably low yields and extended timeline to market. Our foundational model is a large language model that has been pre-trained on nucleotide sequences, including from the NCBI, Human Protein Atlas, patented and other published viral vector sequences, and a proprietary dataset. Upon this extensible base model, we have primary tasks and subtasks that produce the outputs for our vector design predictions and optimization. The greatest value proposition is that FORMsightAI allows cell and gene therapy developers move from preclinical development to IND faster, resulting in drug candidate products that are safer, with lower truncation risk and fewer lab iterations. So today, we want to briefly talk to you about our in silico solutions on how we can design better vectors using AI models. Here are some key challenges that are faced during vector design. And in the subsequent slides, we'll provide you with how our in silico solutions help to address these common challenges. Firstly, it is ensuring that the vector design allows for efficient packaging of the therapeutic gene into the viral capsid, since this is very critical for successful gene therapy delivery. Partially filled capsids can result in reduced transduction efficiency and compromise therapeutic outcomes. Secondly, identifying structural motifs, which can impact the vector's ability to be replicated and packaged into viral capsids, viral particles efficiently. So that is another challenge that is commonly faced during vector design. Thirdly, it is to minimize the immune response to the viral vector, since that is crucial to avoid any adverse events. Then it is to optimize the coding sequence to minimize the risk of premature termination or truncation. And this is important because if the coding sequence is optimized and we're able to get more full genomes, then we are helping to maintain the functional integrity of the therapeutic gene and enhance the efficacy. And lastly, it's to increase the speed at which we can screen vectors so that we can take the lead candidates through in vitro, in vivo testing. So accelerating the screening process is crucial for expediting the development and translation of promising candidates into clinical applications. So in the following slides, we'll go into more details on how FORMsightAI can help solve these challenges. Our AI model is trained to predict the truncation risk given an input nucleotide sequence. So in this graph, the y-axis represents the truncation risk plotted on a natural log scale. And on the x-axis, we have the different vector elements. So when we look at the promoter region, we find these peaks, right? These peaks or hotspots indicate that these are regions in the promoter that are at a high risk for truncation. And this information already provides you with the insight that maybe this promoter is problematic and we should replace it with something that is that would result in lower truncation risk, thereby enabling efficient packaging of the therapeutic gene of interest. Additionally, one of the major readouts that we have is what we refer to as secondary and tertiary structure motifs. So this analysis determines the probability of a given set of nucleotides to fold into a secondary or a tertiary structure, like a hairpin or a G quadruplex. So these structures could correlate with a truncation event like replication fork stalling. So in this figure, we have the probability of a secondary or a tertiary structure formation on the y-axis and the different vector elements on the x-axis. We can see here that in the promoter region, there are these problematic motifs that are being formed at a high probability. And we can also see that there are these shaded regions, right, which correspond or correlate with a predicted truncation event. So this information tells us that these are problematic motifs that we've identified that could actually correspond to a truncation event. Nextly, we also look at CpG island and GC content. So on the left y-axis, we have the GC content of the construct that is indicated by blue line. So basically what we're looking for is the GC content in the biological norm of 50 to 55%. If it's very high, it could result in longer range interactions with other part of the construct or form structural CpG islands. And with regards to CpG islands, that is indicated by the orange line that you see across, we're looking for whether these CG dinucleotides are possible methylation targets, right? So at the 100% mark, that's where we have 100% positive CG dinucleotides that is likely to form a CpG island. And above that, we have a shaded region that is at a high risk for methylation. So areas like the promoter where CpG island formations are required, we typically ignore them. But when we see them in other parts of the construct, specifically like the CDS, we flag them as problematic because we are worried in terms of immunotoxicity. So that's the information this kind of output helps provide. FORMsightAI can also help you optimize the CDS of your construct to reduce the truncation risk. So when we look at the CDS region of this construct that is indicated by the M-cherry CDS here, we see the purple line, which is the initial construct, and pink, which is the optimized construct. And we have the truncation propensity on the y-axis. So we can see that FORMsight optimization of the coding sequence reduce the risk of truncation. And therefore, this helps to improve the manufacturability of the construct. So by performing all of these in silico assessments, FORMsightAI helps you identify the vector designs that have the best outcomes. So this reduces the trial and error cycles and helps to accelerate the development of drug substance. So while having all of these predictions is extremely cool, the first question that comes to mind is, how do these AI models do in the real world? How accurately is our model predicting? So the case studies that we'll be going through today have been done in partnership with leading academic institutes and CDMOs. The initial and FORMsight designed vectors were packaged into self-comp or single-stranded constructs and purified using affinity chromatography. All of these constructs were also assayed for identity using PacBio long-read sequencing. These case studies support the effectiveness of using AI in designing more efficient and manufacturable vectors for gene therapy. So here are the key insights, and we'll be walking you through each one of these insights and show you data in the subsequent sections. So FORMsightAI predictions are highly accurate and reproducible. This is something that we were able to see a very high correlation with between our model predictions and actual NGS data. FORMsight model is also able to accurately identify the optimal combination of vector elements for a specific gene of interest. So let's say you have a gene of interest and you're looking at which promoter would be better. FORMsightAI can accurately predict which of the two promoters would be more efficient in terms of packaging and reducing the truncation risk. FORMsight vectors also reduce undesirable characteristics. We'll show you data that indicates that FORMsightAI optimized CDS reduces the truncation risk, which thereby helps to improve manufacturability. And we're also seeing that optimization using FORMsightAI helps to reduce CPG island formation at the CDS, and this reduces the risk of immunotoxicity. And lastly, and most importantly, we also see that FORMsight designed vectors express well, so we can see mRNA expression as well as functional protein activity. So let's start with the first point, which is that FORMsight, we wanted to check how well our model is predicting. And so on figure 1, we have a correlation plot. On the x-axis, we have the truncation predictions based on NGS, truncation based on NGS. And on the y-axis, we have the truncation propensity based on FORMsightAI model. And we can see that there is a very high correlation between the NGS data as well as FORMsight prediction. On figure 2, we have the overlay of NGS data with our model prediction for 42 constructs. And these data indicate that the truncated drug product outcomes predicted by FORMsightAI are consistent and accurate with the actual NGS data. Nextly, to determine whether FORMsightAI can identify the best set of regulatory elements for a given GOI, we looked at 2 different constructs that have the same gene of interest, but have 2 different promoters, P1 and P2. So on the y-axis, we have the truncation propensity or the truncation risk at the promoter region. And what we can see here is that the truncation risk is increased with promoter P1 when compared to promoter P2, which indicates that our model predicts P2 to be a better performing promoter with reduced truncation risk. So when these constructs were synthesized, packaged, and NGS was run, this is the data that we got. So on the y-axis, we have the read-end counts, and on the x-axis, we have the different vector elements. So what the read-end counts, the way to read this graph is that these peaks that we're seeing indicates that there are a lot of read-ends that are terminating at that particular position. And so when we specifically look at the promoters of these 2 constructs, that's P1 and P2, we see that there are these increased peaks at the promoter region, indicating that there are a lot of truncations taking place. And when we look at the corresponding promoter P2, we see that these truncation peaks are no longer present, right? So this indicates that P2 is a better performing promoter with lower truncation risk than P1, and this corroborated our FORMsight predictions. And so this way, FORMsightAI was able to accurately predict which of the 2 promoters is more has a lower truncation risk and therefore better manufacturability. Nextly, to determine whether the optimization of the CDS using FORMsightAI actually reduced truncated genomes, we used the initial construct and an FORMsightAI optimized construct. And we specifically looked at the truncation risk at the CDS region. So what we can see here is that the optimization of the coding sequence, the initial construct has a higher truncation risk when compared to the optimized construct at the CDS, right? So ideally, the optimized construct should have a lower truncation risk. And now when we look at the NGS data for these constructs, this is what we see. So when we specifically look at the CDS region, we see these peaks for the initial construct, indicating that there's truncation taking place at the CDS. And when we look at the optimized constructs, these peaks have been successfully ameliorated. So the optimization of the CDS actually reduces the truncation risk, and this resulted in a 28% increase in full reads. So this way, optimization actually helped to improve the manufacturability. Nextly, to determine whether this optimization also reduces the risk of immunotoxicity, we looked at the GC content and CpG island for the initial and the optimized constructs. So the initial construct here is in purple, and the optimized construct is the pink dotted lines. So since the GC content at the CDS is hovering at about the biological norm, the optimization couldn't make it, you can see that it is still very close to the initial construct, right? So we are not overfitting or overcorrecting for it. But when we look at the CpG island, we can see that there's this peak from the initial construct, which has now significantly reduced when we use form site optimization. And so thereby we're reducing the risk of immunotoxicity. And then coming to whether the form site design vectors are able to express well, we performed a couple of assays. So here we've done ddPCR to look at mRNA expression. We did immunoblot analysis to check for protein expression. And we also did an enzymatic assay to test for whether these proteins that are being made are actually functional or not. So let's take a look at the mRNA expression that's indicated by the blue bar. So we see that the form site design vectors are able to express mRNA well. This is a open source codon optimized construct. What we actually see is that the form site designed vectors are actually expressing about 20% better than the open source codon optimized construct. When we take a look at the protein expression, we see that the form site design vectors are expressing very well. And whether that translates into activity, we see that when an enzymatic activity using ELISA was performed, the form site constructs are able to make functional protein. We see this with the activity. And they're also about 20% more than the codon optimized construct. So this is one of the experiments where we have the data. We also have another experiment where we saw similar results. And this indicates that the form site design vectors are able to produce biologically active drug products. So I just wanted to reiterate that the biggest value propositions of using FORMsightAI is that it provides actionable guidance on whether the design can be further improved or if the design is already very good, and how quickly we're able to provide this information to gene therapy developers, right? So it saves you considerable amount of time, especially as you're moving your gene therapy vectors from preclinical to clinical development. And they're able to generate vector sequences that have a lower truncation risk, better manufacturability, that are safer, and all of this with fewer lab iterations. So that is the greatest value proposition of using FORMsightAI as you design your vectors. In terms of our roadmap, we'll be continually testing our model with multiple gene therapy constructs, compare the packaging predictions with NGS data, and also perform in vitro assays to test for expression. We'll be working with customers as they take our form site vectors through the animal studies for IND. So that's something that we're very excited about. And as we near the conclusion of our webinar, I want to take this opportunity to extend my heartfelt thanks. Firstly, I'd like to express my deepest appreciation to everyone within our company at Form. Together, we've reached remarkable heights and made a profound impact on the field. I also want to thank our esteemed collaborators at Resilience and UT Southwestern. Your partnership and commitment to excellence have been pivotal in our journey. The lab validation studies we've undertaken together have not only enhanced our products, but also forged strong bonds and a sense of shared purpose. So thank you, each and every one of you, for your invaluable contributions. And with that, I'd like to thank all of you who've attended the webinar and also who will be watching it later on. So a big thank you to all of you. We have a QR code here, which if you scan, you will get an access to the PDF that have all of these results in more detail. So please check that out. And with that, we are open to question and answers.
Yeah, thanks, Anusha. I think one question probably everyone is thinking is how much time is actually saved using FORMsightAI? Like what did you find?
That's a great question. So it actually takes us less than 2 days to perform simulation and optimization. So it takes less than 48 hours to provide the sequences. The main tasks that we have when we get the sequence from a customer is annotation confirmation, predictions for safety outcomes, and optimization with the CDS. And so all of that takes less than 48 hours.
Okay, great. We have some questions from the audience. One first question is, what type of data was used to build the AI model, and how was the data generated?
So I know that the model has been trained on nucleotide sequences. And so the model is agnostic to DNA or RNA. But at this point, we've also specifically trained on AAV-based sequences, which is why we are currently focused on AAV gene therapy.
Okay, great. Does pseudotype impact the prediction accuracy based on the gene of interest?
What I would probably have a follow-up on what exactly they mean by pseudotype.
Okay, so maybe the person can respond a little bit further, and we can move on to some other questions. Can the FORMsight analysis help guide the optimization of codon optimization algorithm?
So basically, right, when we do the simulation, we can already see how well the CDS, how much risk the CDS is towards truncation risk. So when we do the simulation, we can see if there are any hotspots within the CDS that are amenable to FORMsight optimization. So that is something that you can see with simulation alone. And then when we do optimization, we have a lot of levers that we can pull. We can reduce the truncation risk. We can keep the GC content nominal. And we also try to reduce CPG islands. And based on the customer requests, we can tune these different parameters to exactly what they're looking for. And I think that is something that we can work with the customer to figure out exactly what they're looking for. So that's how we can guide the optimization. Yeah.
Okay, great. And I think we have a little bit more clarification. I think when they said pseudotype, they meant AAV serotype. Does that make sense?
Yes, great question. So actually, the data that we've used to train our model does not take into consideration the serotype, because we've trained our model on PacBio long-read sequencing. So the capsid has already been lysed, and then we're looking at the vector sequences. But we have seen with one of our publicly traded gene therapy customers that the serotype also plays a very important role. So if you change the serotype, the truncation outcomes also change. But right now, so capsid in that sense and the serotype is in our R&D. But right now, we're focused exclusively on the vector sequences.
Okay, great. And then lastly, a question came in. Does FORMbio have its own codon optimization algorithm?
Yes, we do. So I know that open source codon optimized is just looking for they're just basically doing codon optimizations. But in this case, we are doing synonymous codon optimization so that we're keeping the amino acid the same. But as I mentioned before, we also have other levers that we can pull, which is like reducing the truncation risk, keeping the GC content nominal, and also the CPG island risk based on whether that's important or not. So those are all the different levers that we can additionally pull. And that's the main difference and how we are differentiated in terms of codon optimization.
Okay, fantastic. So before we sign off, if you're looking to elevate your AAV development programs, please reach out. We're eager to connect. And we'll be sending this webinar as a recording to everyone. So thank you all for your time, and thank you, Anusha.
Thank you so much, everyone.